Okay.
Okay.
All right. Thank you all for coming. It's a pleasure to introduce our speaker today, Mike Sandel. I've known Mike for quite a while. He and I have been going to Southeastern Fishes Council meetings for many years. We talk at meetings all the time, and he did his PhD at University of Alabama, and then was a faculty member at University of West Alabama. And we kind of kept in touch all these years. And I found out at the last meeting I went to with him that he had taken a position at Mississippi State. So I thought, well, this is a good opportunity to have him down, talk about his research. And so that's what he's going to do today. And I'm very intrigued by the title. So I know all our interactions are strictly on fish conservation, and we'll, a little more fish systematics type stuff. So the title is intriguing, so I'm looking forward to hearing about it. Mike?
Yeah, thanks, Jake. Thanks for the introduction. And thanks everybody for coming. I'm excited to be here and talk about some research that if you had asked me when I was sitting in your seat as a grad student, would I be a faculty member presenting on mycobacteria maybe 10 years later, I would have laughed because it was not in the plan. A little bit about myself. I grew up in Ohio, flipping rocks after school, catching minnows and crayfishes and things like that, and really fell in love with biodiversity. Even though Ohio doesn't have a lot of it, it was still my pastime, and that's what allowed me to, make my way through undergrad and earn all the degrees I needed to have this career. And the path took me to Alabama, as Jake said, for graduate school, where I fell in love with genetics, with phylogenetics, evolution, and those types of analyses. Then went on to UAB in Birmingham and learned about genomics and how the genome is actually allowing us to understand the function of all this fascinating genetic diversity that I was studying as a graduate student. And although at UAB I wasn't able to study fish, well, in my spare time I could still do some fish work, but most of the work I was doing at UAB was on human genomics, working on colorectal cancer, heart disease, and things like this. And then later I was lucky to have a faculty position in the state at University of West Alabama, where I got my feet wet as a faculty member and was able to train students to do some of the same things. And my goal in this endeavor to become a faculty member was to bring these skills in genomics that I learned about human disease and things like this back to fishes and back to the rocks that I was flipping and the creeks that I was stomping in as a kid, and to combine these two worlds. So the whole time I was a postdoc, I was thinking, how can I find some problem that's important to human health, to public health, as well as to environmental health? And it turns out I wasn't the only person trying to figure this stuff out. I wasn't the only person thinking about this. There's actually a whole lot of people thinking about this. So this is Earth, and one of the most fascinating things about life on Earth is that it's non-randomly distributed, right? Species diversity has specific patterns across the globe. We know that the tropics have the most species, whether you're talking about the rainforests or the coral reefs, and the Arctic polar regions have the fewest species. And since the days of Darwin, we've been trying to figure out what explains these patterns, what processes are driving the geographic patterns that are non-random that we see in nature. When I came to the South, why were there 24 darter species compared to the 2 species that were in my hometown in Ohio? These are essential questions that you can ask whether you're studying fishes, plants, bacteria, or other organisms. And so, so the work that we do in my lab most of the time is dealing with fishes. We're still a fish conservation genetics laboratory. This is one example of some work that a graduate student of mine is doing on the endangered spring pygmy sunfish. And we use techniques like DD-RAD-seq, double digest RAD sequencing, which is a survey of the genomic variation within the species to look at things like heterozygosity, effective population size, and fixation indices across the range. What you see here is a phylogeny based on, here we go, just did it. A few gene sequences as well as a PCA based on DD-RAD-seq data. And what my student has been able to learn about this endangered pygmy sunfish here at the base of the phylogeny is that there are three very distinct genetic clusters, and they should be managed as independent management units by the Fish and Wildlife Service. So the work that we're doing is informing decision makers who are engaged with the Endangered Species Act and the conservation of freshwater biodiversity. There are species or populations here that have gone through bottlenecks, and they need to be preserved, possibly supplemented with additional genetic variability as well. Another project that's recently been wrapped up in my lab is a survey of the tri-spot garter, another endangered species that's found in Northeast Alabama, in parts of Georgia and Tennessee, but it's a very rare fish that you can think of as a tiny salmon. It moves from large, well, streams about the width of this room to very small streams that you would be able to step across most times of the year. And that small stream is where those fish is reproducing. So it's migrating back and forth between large and small streams. And this is why it's very hard to find this fish. It's very rare, and it's on the endangered species list because these habitats are easily disturbed. If we don't even recognize this habitat as a stream, it's easy for people to fill it in or plow it over or what have you and destroy the habitat. Similarly, my research associate found that this species is also comprised of three distinct genetic clusters. This time, we have inferred that there are some impoundments here that are causing gene flow to be separated or to be interrupted. So our modification of the landscape here has changed the demography of the species. And again, we have three management units that the Fish and Wildlife Service needs to manage independently. We've also done some eDNA analysis, environmental DNA analysis of this species, and found new spots to survey. So new potential streams that are being used for reproduction that we didn't know about before. So lots of cool stuff is happening in my lab. But back to the focus of this talk is really integrating those conservation themes with things that are important to everybody because they affect our health. So as I mentioned before, I'm not the first person to think about these integration questions. The One Health movement by the CDC is really gaining some momentum, and it integrates human health as well as veterinary medicine and agricultural health with environmental health. Because there are so many problems that are rooted, that have the same cause that affect so many different systems. It's really doing a disservice if we're only looking at isolated problems, such as cancer due to exposure to environmental contaminants or radiation effects or things like this. They all should be studied in an integrative manner. Veterinarians should be talking to scientists, and they should be talking to physicians as well. But one of the, I'm going to go back on purpose this time. One of the themes that you'll see with a lot of the One Health projects is that they're focused on terrestrial biodiversity. The environmental health side that you see here in green is primarily dealing with livestock, maybe some other terrestrial animals that we interact with on a regular basis, our pets, but not much is being done underwater. And so one of the goals for my work is to take this concept of One Health and bring it underwater and think about the water systems, the aquatic systems that we interact with regularly. And so the problem statement here is that there's a group of bacteria that affects not just only humans, but also fish. And when I discovered that there was a bacteria that causes outbreaks in wild fish populations that I could study, by stomping around in creeks, and then also could apply that to things that are important to human health, I was really excited. Right? This group is called Mycobacterium, and the genus Mycobacterium also contains the transmissible bacteria that's responsible for tuberculosis. So you're all familiar with the word tuberculosis, but you might have forgotten that it's the deadliest infectious disease we've ever known. For 1 year, in 2021, COVID-19 had a higher mortality rate than tuberculosis. But quickly thereafter, tuberculosis regained that title. So even today, tuberculosis is killing more humans than COVID-19. Importantly, we've been able to slow that disease spread over time with different public health interventions, about 2% decrease per year globally. So that's a good thing. The blue line here shows that it's going down. What's strange though is that the related species of Mycobacterium are also causing diseases. And these diseases are increasing in incidence at about the same rate, maybe even faster than we're seeing a decline in tuberculosis. These are called non-tuberculous mycobacterioses or non-tuberculous mycobacterial infections. And that's what we're talking about today. It makes sense to look at fish as a potential reservoir for these bacteria because they comprise more than half of all vertebrates. You might be familiar with this if you've taken an ichthyology class with Dr. Schaefer, but most vertebrates have fins. They're in the water. And if we're going to look for pathogens that can be contracted across species boundaries, it's a good place to start. And so we're taking the One Health concept underwater and thinking about the fish that we interact with, whether for consumption or for other purposes, such as even your drinking water or places where you're washing your clothes, right? There's a bacterium called Mycobacterium ulcerans, which is the focus of my talk, which causes a flesh-eating infection called Buruli ulcer. It's quite common in Africa and other tropical regions. And if the patient doesn't have access to good health care, the infection can progress to the point where they need amputations. There are occasional mortalities associated with this infection as well. One of the problems and the reasons that you see people with this real extensive necrosis is that the bacteria has a compound that shuts down the nervous reaction so that the patient doesn't feel any pain, even though their skin is literally necrosing and falling off. So these children are not necessarily complaining about their condition, even though it's obviously debilitating. This map here shows the disease incidence. The darker the blue, the more often you see cases. Australia is a more recent set of outbreaks, and some other researchers have started looking at the geographic distribution of these infections and associating them with human migration. And if you know anything about the history of Australia, you know that they are that the colony was founded by outcasts from Europe, maybe criminals and that sort of thing. And so some researchers have associated the outbreaks of Buruli ulcer with things like slave trade and colonization of different continents. It's a very closely related group of bacteria, and the outbreaks have been relatively recent. What's interesting to me is that in North America, we also have a very similar bacterium that's causing outbreaks in striped bass. So this is a sport fish in Chesapeake Bay. We also have our own striped bass here in Mississippi, the Gulf strain. And in Chesapeake Bay, there's a very serious pollution problem coming from the Maryland and Washington, D.C. area that causes water quality to decline. And associated with those water quality declines are algal blooms, hypoxic zones, and lots of fishes coming up with lesions on them. This is obviously important for the fishery. It's a multi-million-dollar fishery because the fishermen can't sell these fish. And the people, recreational fishermen, are just releasing them back into the water. They don't use them, so they'll stop fishing. If this is the only fish they're going to catch, they'll just stop spending money on the industry. So tying these two things together requires a comprehensive look at the environmental health of not only Africa and the tropics, but also North America. One of the more important water quality events that most of you should be familiar with is the Horizon oil event of 2010. And the oil spill covered a very large geographic area throughout the Gulf, shown here, including the entire coast of Mississippi. And some of the ecological effects of this oil spill are that fishes were reported coming up with lesions on the skin after the oil spill. And they reported an increased number of lesions before and after the oil spill. Everything shown here in red was after. So different types of species here had more lesions after this occurred. Another study looked at the red snapper, and the fishes have a dermal microbiome that exists in their slime. So they looked at the microbiome of these red snapper after the oil spill and found that Mycobacterium was one of the groups that was increased in abundance within the red snapper here. So after the oil spill, Mycobacterium became a greater proportion of the microbiome of these fishes. So are these associated? These two studies don't really connect that, but they're circumstantial pieces of evidence pointing in that direction. So we looked at, and others have looked at the genetics of this bacterial group and found that they're very closely related. The fish pathogens and the human pathogens are very closely related, and they differ not so much in the genome, the chromosome, which you see here, but in a plasmid, which if you know much about microbiology, you know that bacteria can carry plasmids, which are extra chromosomal pieces of DNA that can cause totally new functions, totally new chemical reactions to occur. And in this case, the plasmid carries an enzyme that produces that toxin that shuts down the nervous system reaction. It also shuts down the immune system response. So normally, when a human gets a mycobacterial infection, the immune response is to encase it in a granuloma. It's almost like a little tumor that shuts down or entraps that bacterium in a prison of immune cells. This mycolactone compound shown here actually prevents that from happening, and that's why you have necrosis and an ulcer formed instead of a granuloma in this particular bacteria. But the chemical composition of this mycolactone determines whether it's highly virulent in a human or a fish. So the different genotypes of the bacteria determine whether it's going to cause a serious disease in a different type of host species. So we were a group of us, myself and two researchers from the University of Michigan State University, as well as some French researchers who were also interested in this problem from the Institute for Research and Development and the Center for Research and Economic Development from France, all came together and wrote a proposal to NSF to study the ecology of this bacterium in the tropics, where we thought that the ancestor to both groups of Mycobacterium ulcerans evolved. So the fish ancestor and the fish pathogen ancestor and the human pathogen ancestor, it all kind of pointed to this region in northern South America. And we were lucky to be funded in 2019. So we were all celebrating, right, in about September of 2019. And then obviously, we all know what happened in 2020. So our travel plans to go to the tropics were delayed. And we had to figure out what are we going to do now that an infectious disease has interrupted our infectious disease research. We decided to do some literature review and look at the evolutionary patterns that we could find, not just within Mycobacterium, but also the related genera. And then also we went to the coast of Mississippi and Louisiana to do some field work here because we were allowed to travel domestically as long as we took the right safety precautions. And so we adapted, and we're still able to get some research done, even though we weren't in the tropics yet. So backing up and looking at the evolution of these bacteria, Mycobacterium is in a group called Actinobacteria. Actinobacteria is famous because they are actually the group that is responsible for the production of our antibiotics. Most of the naturally derived antibiotics that we take are synthesized by this group of Actinobacteria. In fact, mycolactone, that compound that actually harms us, is a derivative that's very similar in many ways to things like streptomycin. So this is a group that's very important biomedically because it contains these very helpful bacteria, but also the most deadly bacteria known to mankind, tuberculosis. So we wanted to look at all of the species descriptions of these bacteria and see where they come from. can we start to look at the phylogeny of these bacteria and treat their point of origin or their discovery as a trait or a character? And can we kind of look for non-random patterns of their discovery across the phylogeny? So we're grouping these bacteria, over 1,000 species, actually over 1,100 species, up until July of last year, grouping them into their genera as well as where were they discovered. And what you find is that the family containing Mycobacterium is Mycobacteriales, and most of the, or you could say the greatest proportion of Mycobacteriales were discovered from an organism. This could be a plant or an animal, but fewer were found in the environment, maybe a soil or water sample. And then there's a certain proportion that is not reported. We don't know exactly where they were discovered. If you look at the other groups, which are actually the helpful bacteria that produce these antibiotics, most of them were discovered in soil samples or water samples. Only a few were causing any sort of infection in an animal or a plant. And so already, just looking at these two families, we see a bias in where they were discovered. So why is this the case? This group of Mycobacteriales shown up here has an extra cell wall made of mycolic acids. And these mycolic acids are a waxy substance that actually protects the cell from the immune system. When our macrophages try to ingest the bacteria, they actually fail to digest the bacteria and it survives. And it actually just pisses them off. They come out very angry, and that's when they start to cause disease. So they're not actually digested effectively because of this mycolic acid cell wall around their cell. We think that this originally evolved to prevent dehydration. So they can survive in soils where other bacteria might not because of things like dehydration. But it pre-adapted them perhaps for virulence. So that might explain this. So within the organism pile, what types of organisms are they being described from? Most of them are being described from animals, with a very few being described from plants. And then if we break that down further, just looking at this family Mycobacteriales and the ones that were discovered from animal infections, which genera are comprising this pile here? And we see that Mycobacterium, the genus that is responsible for tuberculosis, makes up most of these animal infection groups. So essentially what we're finding here is that there's a non-random pattern of post-pathogen coevolution going on. And that this group is particularly likely to be associated with animal infections. When we look at that genus in depth, there's been some phylogenetic analyses on the genus itself, and they've done some really great work because phylogenomics has allowed us to differentiate and predict disease virulence in many of these different taxa. Where before we were trying to guess based on how fast something grows on a petri dish, whether it would be a pathogen or not. Now, phylogenies allow us to make much more accurate predictions. And so this group here is the Marinum complex. These are slow-growing bacteria. And this clade up here is the clade that produces that mycolactone plasmid. Another part of our literature review was to look at where in the animal kingdom these infections are being reported. And I wanted to look at fish because that's where my expertise is. And if you look at the phylogeny of fishes and the reports of Mycobacterioses, you again see a non-random pattern where most of the infection cases are being reported for a group called Percomorpha, which includes Cyprinodontiformes and the Perciformes. These two groups are predisposed to, they are much more likely to show up in literature as having Mycobacterioses. There are a few other taxa that have had this as well, everything from shads to even some amphibians and a few minnows. So in aquaculture, you can see that the same trend occurs as in wild-caught populations. There's something about this group down here that makes them more likely to be infected by mycobacteria. And if we just take a look at the titles of these papers here, mycobacteriosis in South China within Latiolabrax. This is very closely related to that striped bass that has outbreaks in Chesapeake Bay. So this family of striped bass-like fishes seems to be very prone to infection. Another group that comes up again and again in these literature searches are the pisciliids. The pisciliids are live-bearing fishes. If you do any work in the field, you might have come across the mosquito fish in Mississippi, which is a classic member of Pisciliidae. The mosquito fish and others have been introduced widely throughout the world to fight mosquito-borne diseases. So by through World War II and the Korean War and things like this, the military often took mosquito fish and related species to the battlefield and released them so that their soldiers wouldn't get yellow fever and dengue and things like this, which is another interesting component to this study. So the pisciliids, the live-bearers, as well as the striped bass and their relatives seem to be predisposed to this infection. I had an undergraduate student at UWA who looked at the phylogeny of invasive guppies, which are a pisciliid that has been widely introduced, and compared that to the phylogeny of these bacteria that are now tropically distributed around the world, and found that there's a lot of congruence between the history of this guppy being introduced to places like Australia, Japan, as well as the bacteria. So this doesn't tell us specifically that our introduction of this fish is responsible for the introduction of the bacteria, but it does provide evidence that we need to investigate this further. Shortly after that student finished, I had a graduate student that went back to the Gulf of Mexico and looked at the red drum, which is another species that was being found with ulcers on the side after the Horizon oil spill. Hennigan et al. In 2013 were the first to report this, but they didn't do an analysis of the microbiome of the fishes. So my graduate student looked at the microbiome of this fish in the Gulf of Mexico. These are the sampling sites throughout Florida, all the way to Louisiana. And the results of this study were that we found quite a bit more Mycobacterium in the fishes with lesions, although it was always less than 3% of the total microbiome. But that when we did find up to 3% of Mycobacterium, the phylum level microbiome was shifted in those fishes. So this small proportion of the microbiome might have been having a disproportionate effect on all the other taxa that are on the surface of the fish. All right, and so these two studies were kind of the preliminary data that allowed us to get the funding through NSF to go to the tropics. And like I mentioned, we weren't able to go until very recently because of COVID. So a lot of our work started along the coast, looking at various fish species in the same sort of habitats that my first student looked at, from Alabama over to Louisiana. And in this case, we were looking at not just red drum, but lots of different taxa, lots of different habitats, looking at different salinities and pH, dissolved oxygen, and so on. We use seines, dip nets, and minnow traps to collect our fishes. And this is the initial kind of fish community data that we collected along the coast. So what you're looking at are 3 different pie charts, which represent different seasons at the same site. So in 2020, August of 2020, this sample yielded only 3 species of fish. But in March of 21, we had quite a few more, maybe 10. And we started seeing some positive MPM positive, which means that the fish were carrying that plasmid that we were looking for on these fishes. At another site, this is in Mississippi. This is somewhere around this is somewhere around Biloxi. You see a similar pattern where the fish diversity appears to increase, And then we get a positive in March of 2021 at that site. We don't have enough data here or statistical power to say that fish diversity is associated with NPM abundance or Mycobacterium abundance, but it's pointing in this direction. We hope to get funding to investigate this further. Importantly, some of the fishes that we reported as positive are either within the family Poeciliidae, those live bears, or closely related taxa sharing the same habitat. If you take all of these collection events and put them on a PCA and sort these collection events by their fish community, you can see some similar patterns here. So every dot on this PCA represents a sampling event. The triangles connecting the dots indicate that it's from the same location, just different seasons. And the red dots over here on the upper right indicate the sites that have positive detections of our bacteria on these fishes. And what you notice is that the weighting scores here indicate different taxa and how much influence they have over the community. So the longer the branch, the more likely you're going to find that fish in those sampling events. Gambusia affinis, the mosquito fish, is associated with these positives over here. Additionally, we have other live bearers like the least live bearer and the sailfin molly, which are also pointed in the same direction on component 2. So in that upper quadrant there. So again, we have this indication that Poeciliids are associated with NPMs, not just in literature, but in our own data that we're collecting along the Gulf of Mexico. Now we get to the fun stuff. We actually got to go to French Guiana starting in 2022. We made 3 trips to make up for lost time. Our students actually lived in the Amazon for 3 months, from September through October of 2022. And they were able to collect from a number of sites. We sampled 3 different river systems in French Guiana. This is the Mana River, this is the Sine-Marie River, and this is the Aparague River. And we tried to sample in 3 distinct sections of the river, being at the headwaters, the middle of the river, and then down near the coast. So we tried to cluster these into 3 sections. It didn't always work out perfectly, but that was our intent. Today we're talking about the sections or the samples that are shown in black. We haven't processed all of these samples yet, but we did similar techniques. We collected about 100 fish at each site and ran our PCRs to see if this plasmid is present on the dermal microbiomes of the fishes. Again, we tried to sample habitats of a variety of parameters, salinity, temperature, and so on. And we tried to match them somewhat with what we were doing on the coast so we can make comparisons across continents. In the tropics, of course, we found a lot greater fish diversity. We don't even know everything that we collected. Don't tell customs that. Because a lot of these don't even have names on them yet. They've not been described scientifically, and so we had to take our best guess about what we were actually collecting. I forgot it was being recorded. This is, so far we've had 1 positive out of our French Guiana sample. So NSF invested a lot of money in us to find this bacteria on the fish in the Amazon. Our results have not been spectacular yet. We've only got 1 positive, and it's not on the groups that we thought it would be. This is on a tetra, which you can actually buy something similar to this in an aquarium store if you were to go shopping for one. And this fish actually had NPM on its skin. And so it kind of throws a wrench in our theory about, you know, these non-random patterns that we're seeing in other places. Something different might be happening in French Guiana, but this is a very speculative conclusion because we don't have enough data yet to look at patterns. So we're going to keep processing our samples and see what else comes up. In the meantime, we've got our collaborators looking at the soils and the plants and the water samples and insects and everything else in the environment to see where else this bacteria might be coming from. In the end, we want to know how can we protect the people of the Amazon from getting this infection, and what can they avoid, or how can they change their behaviors so that it doesn't cause a public health concern anymore. While we were at this site, sampling in the rain here, just before it started raining, there was a family that came down and bathed in the creek. This was their bathing area. They didn't have running water, and their children were, bathing in the water. And we found NPMs at this site on the plants. So it is a concern for the people that are directly in the area that we're working in. But very low prevalence overall, whether we're talking about the Gulf Coast or French Guiana. It's hard to find this bacteria using molecular assays. It's even harder to find it using culture methods, trying to grow it on a petri dish. But in the end, my student Alex Rakestraw, who just recently graduated, was able to find 8 new isolates of this plasmid. And now, because he used a sequencing method, which has never been done before, it's always been just looking at electrophoresis results. This sequencing method allows him to put his isolates on the phylogeny of other Mycobacterium isolates. So what we can do with this information now is tell public health officials which isolate is actually present at each site. Is it the one that causes human disease, or is it the one that's more often associated with fish disease? This is the clade that includes all known human cases that, well, not all of them, but the most virulent human cases, the ones where people have to get amputations and this sort of thing. This is the real problem child of the phylogeny. And what Alex found was that there was a silver-side fish that actually carried the human virulent from Mississippi. So it was found in this state. For the first time, we know that there's Buruli ulcer here in Mississippi along the coast. In Alabama, he found Gambusia, which again is the member of Poeciliidae. 2 of them in Alabama had the same clade member. So they were different isolates, but they were very closely related to what we see infecting humans and causing major disease. This clade up here is most often associated with the striped bass. So this is the group of bacteria that causes those outbreaks in Chesapeake Bay, as well as the red drums and other large sport fishes in the coastal regions. Alex found some new isolates, including another Gambusia, mosquito fish, a pygmy sunfish, and another Gambusia in Mississippi. And then you'll notice this one here in red says Pearl River. We actually obtained striped bass, which is supposedly of the native Gulf strain, from the Pearl River near Jackson, Mississippi. This was harvested by a member of the state conservation agency. He showed me a picture of this fish with the lesions on the face, and I said, we need that fish. Like, do not eat it. I mean, you can eat it later, but save the head. Like, no one's been infected by eating fish that had it. So he actually, I think, did eat some of it. But he saved the head, and we were able to find, for the first time, the same mycobacterial isolate that affects the fish in Chesapeake Bay is now affecting fish in the Pearl River, Mississippi. So this bacterial infection or epidemic is spreading, or maybe has been here a long time and we've just never detected it before. But the same thing is happening here in the Pearl River. A third clade that Alex detected a fish positive was from Mississippi. This is another silver side. And strangely, this isolate was related to strains that most often infect amphibians, but have also been found to infect, this is a flatfish, like a sole. So it's infected some fish before, but it's most often thought of as an amphibian pathogen. So that's something new to look at. And this is just looking in more detail so you can see the clades, but it's kind of blurry, so I've already covered this. But this is kind of the synopsis of what we've discovered so far, is that for the first time, we're getting Mycobacterium ulcerans from the Gulf Coast of North America. And for the first time, we're finding the isolate that actually causes human disease in our local environments. The third result is that we're finding multiple ecotypes or multiple variants in the same spot, sometimes on the same fish, which has never been recorded before. Previously in literature, Africa had its own isolate, Australia had its own isolate, Japan had its own isolate, and they didn't see variation at the same site. Now we're seeing that there's variation in the isolates within North America. So this suggests that either other reports are underestimating the diversity that's present, or that we're seeing lots of colonization here that's relatively recent. So maybe these things are actually being transported from other continents to the coastal regions of North America, and it's potentially going to be a public health concern. So far, the only human cases that have been reported in North America are from people that have traveled to these other places. So probably research. People that have come from Africa and Australia and gotten sick after they came home. But that doesn't necessarily mean it's always going to be the case. This is something that I think public health offices or agencies need to start monitoring. Also, our literature review showed that the pattern of infection is non-random. We see specifically there's a group of bacteria that we need to be most concerned about, and it's this group of slow-growing bacteria that's related to tuberculosis. And we see that most of their infections are aggregating in certain types of fishes, or in some cases humans. So these non-random patterns are showing up on both sides of the phylogeny, the host phylogeny as well as the pathogen phylogeny. Finally, in our future directions, we want to look at the ecology of this system a little bit more rigorously. We haven't thought about the trophic network at all. And if you know anything about ecological contaminants, a lot of them can bioaccumulate, meaning that the concentration of the toxin can increase as you move up the trophic pyramid or trophic network. So that predators are going to have a higher concentration of toxins than prey species. We don't know if that's the case with mycolactin. So this bacterium here at the bottom is not only consumed by fishes and attacked by the immune system of fishes, but it's also consumed by protozoans, single-celled eukaryotes that try to do the same thing. Amoebas, for example, and some ciliates will consume these bacteria. And if you know about the ecology of the Gulf of Mexico, we often have protozoan blooms, such as red tide. These are caused by creatures somewhat or similar to Carina brevis that become super abundant. And we don't know if there's any connection between these red tides and what's actually killing the fish. Perhaps Mycobacterium is also involved in pathogenesis of food fishes, specifically the menhaden. Along the coast of Mississippi and Chesapeake Bay as well, menhaden serve as the #1 food source for striped bass, red drum, and a few other species. So during certain times of the year, the menhaden schools are so dense that everything, it's a smorgasbord. And you've probably seen the nature videos where you've got fish coming up from the tuna, coming up from the bottom, and birds diving on the school of fish from the top, right? Everything's eating the same prey. That's what happens with menhaden. And so are the menhaden a key link in this system that then distributes the bacterial infection to all these different predators? So this is something we're going to look at in the near future. And of course, we're going to continue our work in the tropics as well, and hopefully go back to French Guiana with an extension of our current grant. All right, with that, I'll acknowledge all of the people that have helped us, both in South America and North America, and our funding agency, the National Science Foundation. And with that, I'll take any questions. Yes, in the back.
Very nice work. I really like your One Health approach.
Thanks.
So my question is that on the past, we know that there are parasites in pig farms, and now with the mycobacterium, so no sushi at all. So my question is, you look at these bacteria, and you're looking at the microbiota. So equivalent to the human lung mycobacterium. I mean, it's a slow growing, it exposed and resides in the lung, and then it causes like pneumonia and all. I mean, have you seen all the infections in the field? And what was the microbial kind of composition, microbial ecology inside the gill in these victims?
So great question. The question was, in a sense, do the fish have a concentration of mycobacterium in the gill, since we know that humans are predisposed to infection through our lungs with tuberculosis? And the short answer is that with these small fish, we have sampled the gills indirectly. So the whole fish is placed in a tonic solution like acetone or ethanol, and all of the mucus then precipitates off of it. So the mucus that's in the gill and the surface of the fish is all precipitated into one pellet. So I can't answer your question for the small fish because those bacteria may have come from the gill, they may have come from the skin. The larger fish, most of the time, we were sampling just the sides of the fish. So it was a time-limited process. Most of the time, we were looking at fish that were harvested for a tournament, a fishing tournament. And you've got to sample the fish and pass it to the next researcher who might be looking at the liver or something. And you don't have time to do a whole lot of work on the fish itself. So most of it was just skin. We did sample some gills, and those data are not presented, but occasionally we did find mycobacterium in the gills. But the short answer is I don't have any data that suggests that it's concentrated in the gills. So it seems to be that we can find it in either place, but I don't know that it's concentrated in the gills. Does that answer your question?
Yeah. Another question is, there are a lot of these mycobacterium, several species.
Yeah.
I mean, is that only the aquatic fish that is straight up infected with mycobacterium, or some of the other invertebrates, or some of the other, the organisms as well?
Yes, so we do find it. I don't have the data with me today, but our collaborators have found Mycobacterium ulcerans in various insects as well. For some reason, it does seem to concentrate around the mouth parts of the insects. So they can put the whole insect in a solution like we do, and they actually have a hard time finding it. But if they just sample the mouth parts, they find it more often. So whether that's some sort of commensal coevolution going on, we don't know. But maybe it's, for the transmission of the bacteria, they kind of like the mites that live in the beak of the hummingbird, right? They travel down to the flower and back and so on. Maybe that could be happening with the bacteria colonizing the mouth parts of the insects. They also occur on biofilms of plants as well. We can detect them in the biofilm that grows on the outside of plants. So it's not just fishes, although disease appears to be limited to fishes and maybe amphibians, things like this. So we don't see disease in the insects.
So I'm interested in your pattern where you've got a correlation between diversity and recurrence.
Sure.
I'm wondering what hypothesis, what you might think may be the mechanism.
Oh boy, that's a great question. So the question is, do we have an explanation for this very preliminary data suggesting that fish diversity might be associated with the presence of this pathogen? Yeah, I don't have an answer for you. Did you speculate anything while you were thinking? No. Okay. I can tell you that these, even though these are positive samples, okay, so I'm going to speculate now. Even though these are positive samples with more diversity, they tended to come from the same groups of fishes, the Pisciliids. Perhaps the increase in diversity of fishes is bad for Pisciliids, and they're under stress because of different predators and things like this, and that could be increasing their susceptibility to infection. So that's the best I can do. Okay. Yes.
When I think of like tropical diseases that come from the tropics, it always comes to mind that environmental conditions and the temperature and that kind of stuff might have an effect on the spread of the disease. I want to know if you think, or if you guys looked into maybe with climate change, increased temperature of waters, that might be why we're starting to see prevalence like in northern countries. Maybe the bacteria just surviving water for long time to affect this issue.
Excellent question. I'm going to repeat it real quick. All right. So the general idea is that, can we look at climate change as a reason for this tropical pathogen expanding its range into non-tropical regions? Are you looking for graduate school or postdoc or something?
I'm starting grad school in the fall.
Missed the boat. All right. So that's exactly what we want to do next. We want to look at, there's a specific climate change call from NSF, and we want to look at whether this could be associated with that. So they do tend to like the 35 degrees Celsius range. And we hear about tropicalization of North America all the time now, whether it's fauna or just flora. So yeah, we want to look at that. I don't have a great answer for you, but if we start seeing human infections spreading from Florida up to New York, then there's your answer. Yeah. More questions?
More questions? All right, let's start again.